Hi, everyone. This is going to be a series of three videos that all have to do with cell division. And in this particular video, we are going to be talking about DNA and chromosomes. And the reason for this is because DNA is a super important molecule that contains all of the instructions for everything a cell needs to make and build. So these instructions, which are found in the DNA, are fundamental in order for a cell to be able to function. And you might be wondering why we're talking about DNA when I said this was a set of videos about cell division. But we're going to start with this one because as we're thinking about a cell's life cycle or what the different stages that a cell goes through, starting when it's formed and as it continues to sort of live on, the DNA in that cell is going to look different depending on the stage that the cell is in. So we're going to be paying particularly close attention to what the DNA looks like at different life stages of a cell and sort of knowing what characteristics to look for when we're thinking about DNA. I think it'll be helpful as we're thinking about the cell cycle and mitosis or cell division. Okay, so there are two big questions that I want you to ask yourself when you're looking at DNA or looking at the DNA in a particular stage of the cell cycle. And the first question is it packed up tightly or is it unwound and loose? These are the two main forms that DNA can come in. So if the DNA is packed super tightly, then we call that a chromosome. And this example might look a little familiar, like in the shape of an X thing that people usually think of when they think of DNA. So we would call this form of DNA a chromosome. But DNA can also be sort of like loosey goosey. People compare it to maybe a ball, like a plate of spaghetti, just sort of loose and unwound, like a tangled up mess, maybe like an unraveled ball of yarn or something like that. So when the DNA is in this form, instead, we call the DNA chromatin. So if it's wound up tightly, then it's a chromosome. If it's unwound and loose, we're calling it chromatin. And then the second question that I want you to ask is the DNA a single copy or is each strand of the DNA replicated? So, this is another big difference for how DNA can look. And in some parts of a cell's life cycle, it exists just as a single piece. So, this is one molecule of DNA, one long strand. And each part along this molecule the DNA is unique. There's no parts of it that are copied or duplicated. But at certain stages of the cell's life cycle, the DNA gets copied. And so we end up with two identical copies of the same information. And this makes sense if we're thinking about the fact that the cells are going to need to divide, and that's what this set of video lectures is all about. So, if a cell is going to divide, it's going to need to give its genetic information to both of its offspring cells. And so, at a certain point in the cell's life cycle, it's going to copy its DNA so that when it divides, both of its offspring cells can get the full set of instructions, the full set of genetic information. So, our two questions about DNA are: is it packed up tightly or is it unwound? And is it a single copy or is it duplicated or replicated? And we're going to explore both of these in slightly more detail in the next couple slides. Okay, so this is just to reiterate the point that DNA can be loosely packed. And this is a diagram showing that sort of canonical looking chromosome that maybe you're used to imagining when you think about what a chromosome looks like. And you can see that it all starts out with these long strands of DNA with that classic double helix structure. Um, and then these pieces of DNA are coiled around certain structures, and then those structures are coiled even further upon themselves, and then coiled up even more to make up this chromosome. And we call DNA supercoiled when it's in the chromosome form. And that's because it's really tightly packed together. And then when it's more unwound and sort of unspooled, if you will, then that's the chromatin form. And you can think about this in the sense of maybe thinking about what the cell will need to be doing with the DNA. And that's going to kind of inform whether the DNA is loosely or densely packed. So it's almost like that when the cell is just hanging out and it needs the instructions in the DNA to be accessible, then you don't want it to be tightly packed away, right? You could think about your clothes maybe in your closet if you want them to be accessible to you in your day to day life. Like if you want to put on a certain t shirt, um, then you're not going to want to have each and every t shirt that you own packed up into a tiny little suitcase. That's not super practical. So when the cell needs to access its DNA, when it needs to use those instructions that are in the DNA, it's going to have it more unwound in this chromatin form, which is more open. But if the cell needs to be moving around its DNA, sort of like if it's shipping it off to two daughter cells and it needs to keep track of all the pieces of DNA that it has, then it's going to move into this more condensed, densely packed chromosome form. And that just lets the cell keep track of where all the pieces of DNA are, and it lets it move it around more efficiently. Okay. And then the second bit that we were thinking about is whether DNA is going to be replicated or unreplicated. So here we're showing a single unreplicated chromosome. That's right here. And I was saying before that if a cell is going to divide, it needs to copy its DNA. So, if we're making two cells out of one cell, then we're going to have to double how many sets of instructions that we have. And we'll end up with what's called a replicated chromosome. So, this is a real picture here. And this is a replicated chromosome that's shown. So, anytime you see this X shape, that's going to be a replicated chromosome. Um, so, there's going to be like one copy of the instructions on one side, and then an exact copy, the exact same information is going to be on the other side. And in the middle, where these two regions are, or where these two sort of sides are joined, that's called a centromere. So a centromere is here, or if we want to look at the diagrams, it's sort of like right in the middle where the two pieces are held together. So centromeres are just the regions in the middle that hold these two copies of the chromosome together. And wait, we're not done with the grammar yet or the vocabulary. So we've got a couple more terms to define. So when we're thinking about what to call these two copies of the chromosome, we call them chromatids rather than chromosomes. So instead of saying that we have two chromosomes here, we would say we have two chromatids because they're in the copied form. And specifically, if we're thinking about two copies of the exact same chromosome, like we are here or here, we're going to call them sister chromatids. So these sister chromatids are exact copies of one another, they're exactly the same. And so you can see in this diagram the two arms of the sister chromatids, which are identical, two copies of the same chromosome. And then when they get pulled apart from one another, so the one on the left getting pulled apart from the one on the right over here, um, so once they get pulled apart, in order to go into the two daughter cells, now they're not chromatids anymore. They're on their own, so we can go back to calling them chromosomes. So they go from chromatids to chromosomes once they get separated. So chromosomes get replicated to become sister chromatids, which are held together at the centromere. And then when they're separated, they become chromosomes again. So, to recap this video, our questions are replicated versus unreplicated. That's one big question. So, here would be unreplicated, and here would be replicated if we made a copy of it. And then the other question that we talked about before was, is the DNA loosely packed, in which case it's chromatin, or is it densely packed, in which case it is a chromosome? Okay, so that wraps it up for this first video in our mitosis or cell division series.